Welcome everyone. Thank you for joining us on this penultimate, hopefully what is almost a wrap up of our speaker series this year in lieu of our symposia in this crazy 2020. Today we're going to be hearing from Dr. Carmen Bertoni, who serves as NDF Scientific Director this year. And before that, I want to make a quick announcement. Obviously, the same usual announcements to please keep your microphones muted if it's all possible, or else we'll do it for you. Please do write your questions in the chat box as they occur to you, and at the end we will have an interactive Q&A session. But before I begin, I want to make a big announcement that you may or may not have seen on the news, but it's about to hit big, and I wanted you guys to be the first to know that as of Tuesday, September 1st, we are going to be welcoming possibly one of the foremost physicians and principal investigators on GNE myopathy from the NIH. Dr. Nuria Carrillo will be joining the NDF team of staff as of Tuesday, which we find to be a wonderful, wonderful victory. Nuria, I know you're on. Could you turn on your camera just really briefly? I don't know if you're there, but just in case you are, maybe just say hi, everyone.
Well, I'm really honored. Thank you for the introduction. I'm really honored to be part of NDF starting next Tuesday. I have been honored to work with patients with GNE myopathy over the last 10 years, and I cannot wait to continue on this journey together with all of you.
Wonderful. Well, it's lovely to have you, and I think this is a particularly important call for you to listen in on, because it is genuinely a wrap up of what Carmen has worked so diligently alongside the rest of the Scientific Advisory Committee and myself to pull together in terms of all of the science that we are funding and the reasons why we're funding it. The gaps in all of the requirements for the FDA from the letter that we got 2 or 3 years ago explaining what we need to do in order to pursue gene therapy and genetic treatments in general. So without further ado, I'm going to turn off my camera and I'm going to turn it over to Dr. Bertoni. Dr. Bertoni, take it away.
Thank you, Lale. Thank you to the NDF for the kind introduction. Thank you for giving me the opportunity to talk today. And thank you for all of those that are tuning in and will have a chance to go through this presentation. What I want to explain through the next 45 minutes or so is the strategy that the NDF is taking to make sure that what we are doing is the most effective program that we can build toward developing a cure for neuromuscular disease, in particular GNE myopathy. We're going to go through a quick introduction for those of you that might not be familiar with the disease, and we focused then on the programs that NDF is funding. And I hope this can serve as a wrap up of all the talks that we have had the opportunity to hear through this summer, and making perhaps a better understanding of why we're doing this and what for. Oops, I'm sorry. So what is GNE myopathy? GNE myopathy is a genetic disorder that is characterized by progressive loss of muscle function, and together with the muscle function, obviously, there is a progressive loss of activity. It was identified and described by several investigators around the world independently, which has led to multiple names for the same disease. And here are some of the names that were used in the past. The genetic of the disease, we know that is caused by a mutation in the GNE gene. And how is inherited? It's an autosomal recessive disease. That means that both parents need to have at least one copy of the disease to transmit the disease to their kids. And if both parents have one copy, you will have only a 25% chance, 1 out of 4 of the kids that will develop the disease, so we will inherit both mutated genes. 50% will be carrier for the disease, and 1% and 25%, 1 out of 4, will inherit the healthy copy of the gene. And this is why it's so important to do a pre-screening for the disease to know if the parents could potentially be a carrier. How is GNE myopathy distributed around the world? We know that there are cases throughout the world. Currently we know that there are approximately 1,000 patients that have been identified. The majority we know contain the Persian Jewish mutation. There are other groups in Japan, and then there are other mutations that have been identified in other populations: Asia, Europe, Middle East, Australia, and North America. We know that approximately 40,000 patients should be present around the world, but we only know of about 1,000. And then goes back to the point of why it's important to do diagnostic screening for GNE myopathy. This is the structure of the GNE gene. There are multiple what we call isoforms, so different version of the GNE gene, which translate for different proteins. Here, what you see is just the two most prevalent form. And also what you see is the GNE gene is composed of about 13 exons. And here, what you see is the distribution of the different mutation that has been so far described in the literature around the world. Those mutations, they appear to be located in a specific population, so it sounds like there is an ancestral inheritance of the disease. What is GNE myopathy? What is the function of the GNE gene? Well, the GNE gene is an enzyme that we know, and we've seen a beautiful presentation from Mariam describing the function of the GNE gene. But what it does is basically converts UDP GlucNAc into MANAC6 phosphate. And when the GNE gene is missing or is non-functioning properly, what we have, we cannot produce the downstream sugars and the sialic acid, and this in turn results in hyposilation of the glycoprotein present at the membrane. Sialic acid is shown here, is at the membrane of the muscle fiber, and what you're seeing is the sialic acid located on those long sugar chains at the membrane. This is in a normal individual. In patient with GNE myopathy, the amount of sialic acid present in sialation is markedly reduced. Let's go to the therapy development and what it takes to go from the bench to the bedside. So there are 5 major phases or tiers. The tier 0 is the most important, is the one that basically relies on basic science to do preclinical and animal studies, and is necessary to understand and define the pathology, the mechanism of action, the target, and the validation of the therapeutic approaches. Once those parameters have been identified, then we can go and move into what is called a phase 1 clinical study. We identify a target, we develop a possible treatment, and then we test this treatment into patients. A phase 1 study is usually a proof of concept studies where there's very small number of patients between 5 and 10 are tested, and the scientists are looking and clinicians are looking at safety and how the the target or the therapeutic approach distributes is called pharmacokinetics and pharmacodynamics of the potential drug that is being tested. If it's deemed safe to use, there are no side effects, then we're going to move into a phase 2 and phase 3 clinical studies. Those phase 2 and 3 clinical studies are safety, efficacy, and selection of the dose to be used for future studies, and usually involve the use of a larger number of people, but still remain within very contained. And if everything proves to be safe in phase 2, then it's moved into phase 3 with a larger number of patients. And finally, if proven efficacious, then the therapy goes into market. And the FDA in the United States, but there are other regulatory mechanisms in Europe, for example, the EMA in Europe, if they approve it, then is commercialized and it becomes available to all patients. Once approved, there is a final stage, which is called the phase 4 post-marketing, where again, all the side effects are being tested in all the patients that are receiving the medication through their clinicians and neurologists. The cost of each step increases over time, and when we get to the stage of conducting phase 2 and 3 clinical trials, we are talking of many million dollars just to do the testing. And this is because everything needs to be controlled, checked, double checked, and there are additional tests that are imposed to make sure that it moves into subsequent step and then commercialization. By the time a drug is approved, we already know that it is substantially safe to use, and that's the reason why it can go into the market for the final stages of clinical development. I want to give a perspective of what we are right now and what has been achieved. If we look at the graph, the first GNE myopathy patients were described in the early 80s. So we're talking 1981, 1982, and as I mentioned, there were multiple groups describing the same patients and calling different names. The genetic and molecular studies have begun as soon as the first patient were identified. However, we have to wait until 2001 to identify the gene GNE as the gene responsible for causing the disease. And this has been a real breakthrough because at this point, many of the mutations that were previously being described could be consolidated into one disease. We have to wait until 2014 until all the different syndromes and names were consolidated into one, which is called now GNE myopathy. As soon as the gene has been identified, then scientists were able to move into the production of animal models and begin their testing. In terms of clinical studies, we have had already several clinical trials that have been conducted. The first one was done by the NIH in 2005, 2006 with the intravenous immunoglobulin injection. There were only 5 patients, that there was marginal effects observed and therefore did not move into phase 2 or 3. We have the group from Tohoku University testing the sialic acid therapy. Again, that one was stopped at phase 1. The two most important are the clinical trials that are currently being conducted at the NIH with MANAC, and the clinical trials that everybody know was conducted by Ultragenyx. The Ultragenyx trial was ended in 2018, but is the only clinical, the only therapy that has been able to reach phase 3 clinical trials. And we are all waiting for to see the results that we're going to get from NIH for the phase 2B of the MANAC replacement therapy that should be completed by 2023. And you had a beautiful presentation from David explaining the characteristic of this latest clinical trial. Now, we know the Ultragenyx unfortunately produced very disappointing results. I want to go over a little bit the Ultragenyx trials, because I think we learned tremendously from the trial. Everybody knows the Ultragenyx trial used a standard release form of sialic acid. The adverse events were generally moderated. There were no safety concerns. There were great results obtained in phase 1 and phase 2. And especially in phase 2, there were a significant improvement in muscle strength relative to patients that received only the placebo. However, when we get to the phase 3 studies, what we saw is somewhat not so great results. Sorry. And what happened was the phase 3 did not meet any primary or secondary endpoints, and therefore it was discontinued by Ultragenyx. So we have also, we have to say, the Ultragenyx has several other programs that were more promising. So ultimately they decided not to continue because they had other promising approaches that were trying to test for other diseases. Again, this is also related to how much it costs to get into phase 3 studies and whether it's worth or not to continue. But I think the phase 3 study, and this is why I want to spend a little bit of time on the Ultragenyx results. I think the phase 3 study have taught us a lot. And I'm gonna show you what are the results that were achieved by Ultragenyx at the end of the phase 3 study. This is an article that was published last year and is readily available on PubMed. This is the primary outcome measure. So what they were looking in terms of bringing to the FDA to receive approval. And when you look at the results, the line in blue is the muscle strength achieved in a patient that received the standard release form of sialic acid, and the one in black is the placebo control. What you're seeing, there is some effect achieved in muscle, and to some extent the effect is quite drastic. You can clearly see the difference. The problem and the reason why the trial was discontinued was that these differences were not considered significant. When Ultragenyx analyzed the results and tried to give an explanation, and what they have come up with the possible reason for the failure was the variation in the population and size that were used between the phase 3 and the phase 2. Remember, the phase 2 gave very encouraging results. The treatment period needed to detect a significant effect might have been too short. In other words, a patient received a standard form ACE-ER for 48 weeks, and they were thinking maybe they needed a longer period to see a significant difference. But the most important, and I think this is what makes, we're going to make a lot of sense at the end of my talk, was the lack of valid outcome measures, biomarkers that they could use to show improvement. We are relatively the new disease. We don't know very well the disease. We need to establish ways to really come up with the valid outcome measure. And the other reason could also be the difficulty in optimizing the dosage. And that relates to the fact that we really don't have much to work with other than testing a possible drug directly into human beings, which limits the number of patients that we can use at every single time. So what has the NDF done, and what can we do to avoid another failure like the one we just experienced with the Ultragenyx? So what the NDF has done through the leadership of Lale, which I remember, I can emphasize more, she has done tremendous work for the NDF. So the NDF has assembled a very strong team of clinicians, scientists, including lawyers with experience in various aspects of clinical development, including licensing, manufacturing, commercialization of drugs, but most importantly, on the scientific aspect of what it needs to be done. This group meets regularly. We discuss about key aspects of therapy development, including therapeutic technologies that are most likely to work for GNE myopathy, and what are the state-of-the-art of the technology currently available. The state-of-the-art changes continuously, and there are new technology being developed every single day. And then the areas in the field that needs to be better studied and understood. Once we meet and we discuss, we take decisions that together in a truly collaborative manner. And I can tell you the discussions are often very lengthy, and we start from each of the scientists and the members of the SAC with a different opinion, and by the end of the discussion, we try to reach an agreement, and normally we are able to do so. And once we agree upon a possible strategies, then we make recommendation that are then passed to Lale, and we try to work to solve the problem. So what are the key points and the recommendation that the SAC has made to the NDF? It is clear that the late stages of clinical testing into humans, and we're talking about phase 2, 3, and 4, and ultimately the marketing of the drug for GNE will require the partnership with pharmaceutical companies. There is no way that NDF can raise sufficient funding to take a drug from the bench to the bedside. So what do we need to do? We need to de-risk the technology. In other words, we need to make sure that we identify the good strategic uses of all the technology, and we make sure that by the time we reach the stages of forming partnership with pharmaceutical company, we have everything available to make sure that the phase 1, 2, 3, and 4 clinical trials become successful. How do we do it? We identify therapeutic approaches and targets likely to achieve sustained beneficial effects in muscle, and then we sponsor projects that will be aimed toward that goal. We need to establish and enrich biobanks and patients registry to understand disease progression. As I mentioned at the beginning, we only know a very minimal fraction of the patients that have GNE myopathy. The more patients we can recruit, the better we can understand the disease. We certainly need to generate robust animal models to optimize the therapy, and that's the reason is that what we have seen with phase 1 through 3 of the Ultragenyx, they really didn't have much to go with in terms of animal models. So it was difficult even for Ultragenyx to determine a dose to be used in patients. And we have a very limited number of patients to choose from. In phase 1, I remember we usually start with 5 and then we go up to 10, 15 patients at the most. Once we have done the proper testing in the animal models, we need to be able to demonstrate that the therapy works. And we need to identify biomarkers as well as outcome measures that can we use in the clinic to assess the efficacy, so that when we go at the end of phase 2 and 3 to the FDA with the results, we can clearly demonstrate that there was a significant effect, and that based on the biomarkers and the outcome measures that we were able to identify. So let's go to the first point, identification of therapeutic approaches and targets that are most likely to achieve sustained effects in muscles. So far, the NDF and the SAC has recommended 2 major strategies. One is the gene therapy, and one is the enzyme replacement therapy. The gene therapy uses basically is aimed at correcting the genetic defect by introducing either a new gene or correcting the mutation in the GNE gene. The enzyme replacement therapy instead is focused on replacing the enzymes that either lacking because the GNE gene is not working properly, or they are downstream the pathway. And those are generally MANAC, MANAC 6-phosphate, sialic acid, or sialylated glycoproteins. Let's talk a second of the gene therapy using adeno-associated viral vectors. You have previous talk that have been quite clear on how the technology works. What we know is that the gene therapy using AAV vectors have been quite successful in muscle disorders, and they've already been approved for muscle disorders. Because they have been approved, is more likely to have that we would be able to move it forward into marketing and commercialization. The viral vector, what we are using is just the shell of the vector, the part of the vector that contains their genetic information is completely removed, and instead we are putting in a new gene. The new gene is then incorporated into the cytoplasm, and from the cytoplasm goes into the nuclei and then eject the gene of interest. In this case, it would be the GNE gene. What are the pros of using these technologies? It's applicable to all GNE patients independently of the mutation that they have, can achieve sustained levels of expression in muscle. You basically can control how much protein you put based on how many copies of the gene you want to deliver to, and you are delivering to the muscle. It's already approved for other diseases. The cons, that is some people might have already immunity against the vector used to deliver the GNE gene. And what this causes is that some people might not respond to this type of therapy. I have to say, however, that this is a problem that is common to any form of disease. Those are vectors that most of the people have been in contact with in a previous life, and it's been estimated that, for instance, in Duchenne patient, about 10–15% of the kids have already developed antibodies against those viral vector. For GNE myopathy patients, for which the disease manifests at later stages, the chances of being in contact already with this vector are higher, and there is an estimate of approximately 50%. However, this is a transient vector that is only going to be used, and there are ways to temporarily immunosuppress the patients before before injecting the virus, so that when it's introduced into the patient, the immune system of the patient does not recognize it. And we're only going to need to do this for a short period of time until the viral vector goes into the cells, and that's what we need to do. The immune response is usually generated against the shell of the vector, not its content. The other problem is the dose to be administered. That needs to be clearly defined before we can even conceive the idea of going into a clinical trials. Because GNE myopathy patients are generally adults, they are likely to require the bigger dose than what has been currently been tested in other diseases like Duchenne in kids or SMA in infants. And that will require to scale up the manufacturing and the production of the vector, which could increase, is likely to increase the cost of production to the stage where they could be too expensive to move forward. We are going to have soon a talk from Dr. Martin. He's going to go through this technology in more detail, and we're all looking forward for his talk. The other approach that the subcommittee has recommended to test is the gene editing technology. Many of you have heard about the CRISPR-Cas9 system and how this technology, what the technology does, instead of introducing a new gene, it goes directly into the faulty gene and repairs, corrects the mutation. What are the pros of this technology? Does not introduce exogenous gene. In other words, we don't need to worry about having too many copies or too little copies because it's correcting the genomic defect at the internal level. The GNE gene remains under its own regulatory mechanism, so we don't have to worry about regulating the expression of each copy, and correction remains throughout the life of the muscle cell being corrected. And we know that, especially for muscle, the turnover of the muscle is very slow, so could potentially last for many, many decades once introduced. Sorry. What are the cons? Because the correction occur on at the genomic level and on the mutation that each patient has, obviously we need to optimize the technology for any of the specific mutations that have been described so far. And there's going to be a hierarchy of which CRISPR, which gene editing approach is used for which population, based obviously not of preferences of who we like and we don't like, but based fundamentally of how many patients we have with that specific mutation. There are some issue of specificity and off-target effects of using these technologies. What we have seen is that yes, it does correct the gene defect, but it also seems to be recognized in sequence that we don't want to touch. And this is obviously needs to be optimized, but because there are so many people working on this technology right now, the issue of off-target effects is likely to be solved before we get into the stages of going into clinical trials. It's not been approved for any other of the muscle disorder, although many people are working on using the CRISPR for Duchenne and other neuromuscular diseases. So again, we think that by the time we reach the stage of going into clinical trial with this technology, there are going to be diseases that have already been approved for the use of CRISPR, making our job a lot easier. And we're still far away. While the AAV vector has been tested, and we know a lot about AAV vectors, and is likely to be used in a shorter period of time. Translation into the clinic for GNE myopathy using the CRISPR might still require several years. Again, if you want to know more about the CRISPR technology, we have had two beautiful presentations from the LEC lab, and I really encourage you to go back and watch those again. Enzyme replacement therapy, as I mentioned before, we can choose several different targets. And what are the pros of using enzyme replacement therapy? In theory, we can just ingest, like a pill, each of the target that we want to incorporate into the muscle. The toxicity and the side effects can be controlled a lot easily because all you have to do is just stop taking the pill. They are easy to produce in high quantities and at a relatively low cost. Most of the manufacturing now can be done overseas. India is a large producer of many of those compounds, and the cost is affordable. The dose to be administered can be adjusted depending on patient tolerability and the side effect, as opposed to gene therapy, where we most likely are going to go with one dose and one dose fits all. The beauty also is that can be used in combination with gene therapy. We can take a pill and have an injection of AAV or CRISPR-Cas9 vectors and still get double the effectiveness. The cons, because it's a pill, patients will have to take the pill every single day for the rest of their life, and does not target specifically the muscle and might require high doses of compounds to be administered multiple times a day. Many of you are already familiar, those of you that are taking Manic, how many times a day you will have to take it and how often. Again, if you want to know more about the enzyme replaced therapy, we have beautiful talks from Mariam and David, and I didn't explain more in detail about another project that the NDF is funding, and that is Dr. Yoshioka and Dr. Nishino. What they're doing is they are trying to determine if there are drugs that are already approved by the FDA and that could increase the level of sialic acid present into muscle. And the project has just started a couple of months ago. Dr. Yoshioka, many of you know her. She's a patient with GNE myopathy, and she's highly motivated. She's currently working with Dr. Nishino to try to determine if there are drugs that are already approved that we can take and give to patients. And we're hoping that she will be able to present very soon results, which I anticipate will be quite exciting. The other aspect that the SAC has recommended is to establish and enrich biobanks and patient registry. We need the patients, so we need to have sample from those patients so that the scientists can study and better develop a cure. The biobanks and patient registry help researchers learn more about the disease. This is incredibly important for diseases like GNE myopathy that are called ultra-rare diseases, where you really have to work only with a small number of patients that are known to have the disease. The biobanks make it easier for scientists to get the data and sample they need for research. And I would encourage any of you, if you have not done so yet, to please go on the NDF website and register for our patient registry, and it's totally safe. Information are kept confidential, and before you sign up and you volunteer to provide information about your health and tissue, please read carefully the rules and make sure that you understand it. But I can tell you this has been developed together with under the following the laws and the regulation that are implemented through IRB, or Institutional Review Board studies. Animal models to optimize therapy before we go into clinical trials. So why are they so important? They are definitely used only for the initial stages of clinical development in the preclinical studies. But they are important because they help us understand the dose to be administered to have an optimal effect before we go into patients. It helps us to optimize the therapy in terms of choosing which therapy is best to move forward, which one is the more efficacious. Help us discover biomarkers and also allow us to study toxicity as well as develop valid outcome measure to obtain marketed approval. Who are we funding? So what are the animal models? I explained at the beginning of this talk that since the identification of the gene, the scientists have tried to develop animal models, and there are currently 3 animal models. And I'm not going to go through the description of each of the specific of this animal model, but I can tell you that so far none of the 3 animal models that have been developed are really optimal to test gene therapy approaches in GNE myopathy. Likely for us, over the past 10 years, there have been several new technologies that have been developed that are able to specifically knock down or eliminate the expression of the GNE and its function into muscle, thus avoiding some of the problems that we had in the past with those mice, where they, if you abolish the expression of GNE, you won't be able to have a mouse because they die in the womb or they die a few days after they are born. And the NDF currently is funding the generation of 2 new mouse models, one from Dr. Stella Rosenbaum and one from Dr. Martin, so that we maximize the chances that by the end of the next couple of years, we have a new model to study and optimize all the parameters that we need to have before moving into testing in human. The second animal model is a different model. It's a zebrafish model. And the zebrafish are quite useful. They are very easy to grow. They can grow in a petri dish. You can actually grow thousands of them in a very small space in any lab. And they're useful because they can be used to screen compounds capable of restoring GNE function. They can help us understand and optimize the dose needed to achieve an effect. We can study and understand and identify biomarkers that affect the function of the muscle following GNE knockdown. We can also identify genetic modifiers and develop outcome measures. And I am pleased to report that the zebrafish model has been generated and is currently being shared among all the investigators who are interested in looking at that. And we have already Dr. Weslider and Kelly Crow. They are testing. They are about to receive the zebrafish model for their testing. One of the questions that often comes up is, do we really need an animal model? It's not absolutely necessary to have an animal model to move into clinical trials. However, without the model, we are going to risk to miss some key points, and we are risking to go into a second failure like the one that was achieved through the Ultragenyx trials that reached phase 3 and then had to be terminated. We need to really do our diligent work and make sure that we cover everything we can possibly cover, including testing into the model before we go into phase 1 study. And if we really cannot do that, then we can proceed without the animal model, but we certainly need to exhaust every possibility that at least we have done the work. If you want to know more about what we are funding in terms of animal model, again, I refer to the beautiful presentation that Stella gave a couple of months ago, and as well as the one that we hope will come soon from Dr. Martin. Oops. The other aspect, the other recommendation made by the SAC member is we need to identify effective and valid biomarkers. They are going to be used in clinical research to identify protein signature that can predict outcome and responses. Why are they important? Because they might provide insight into the biology of the disease. We need to understand better the disease. We can effectively stratify patient population once we have a therapy that we want to move into clinical trials, so that we avoid possibly one of the problems the Ultragenyx are, that the biodiversity and the heterogeneity of the population that was enrolled in the clinical studies. And obviously, they advance our understanding of the disease and biology. We could identify novel drug that could be targeted, some of which might already been approved by the FDA, and therefore moving those drugs into the clinic would be a lot easier. And it can be used to monitor the effect of the therapeutic intervention. And this is the last point, is critically important. If we cannot demonstrate that a therapy is efficacious, then the FDA will never allow us to move into marketing. And without those biomarkers, it's also much more difficult to convince pharmaceutical companies to invest in the late stages of clinical development, phase 1, 2, and 3, 4 final commercialization. Again, this is a summary on all the tiers that we have in the development of a treatment for GNE myopathy. The biomarkers are going to be used throughout all the different stages. What type of biomarkers are we trying to identify? Well, you have heard the talk from Marian and Dr. Crow and Kelly. One of the most promising is probably the sialation of muscle membrane. So the presence of sialic acid in a patient. We know that patients with GNE myopathy don't have or have reduced amount of sialic acid, and you can see here from the staining of a muscle, what you're looking here is the muscle fiber, this is the membrane, and what you're seeing in a patient is that this green staining around the muscle fiber, which is the membrane of the muscle fiber, is diminished or absent in some cases. Who are we funding? Kelly and Mariam, they have presented their work before. Their presentation is quite clear on how we're going to do it. In addition to that, Mariam is at NIH is trying to develop new muscle biomarkers that we hope they'll be successful, the studies will be successful, and within the next few years. Another approach to identify biomarkers and possibly a good outcome measure is membrane repair. We know that from studies that have been conducted through one of the grantees, Noah Weslider, we know now that muscle membrane isolated from GNE myopathy patients are less amenable to repair following an injury. So we could potentially use this model to test how efficacious a drug that we're moving into, or we want to move into clinical trial is, and we can use it in both at the animal model level as well as in patient after isolation of tissue sample from patients that have been treated with the potential therapy. The other advantage of testing this system is that MG53, which is a protein that we know is involved in repair, could potentially become a new target for therapy in the upcoming years by, for instance, expressing or optimizing its expression so that patients that receive treatment, independently of the type of treatment, could also receive MG53 to improve the overall muscle membrane health and potentiate the repair of the membrane in response to daily activity. Again, we had a beautiful presentation from Dr. Noah Weslider at Ohio State University, and I encourage everybody to please do watch again his presentation if you want to know more about the technology that is being used. Finally, identify biomarkers, genomic and proteomic analysis. They're currently being conducted in collaboration with Maduri, Perkin-Ebner, and Monco at Yale University. And this will use samples isolated from patients, and I cannot reiterate more. If you are willing to participate, please donate your sample. This is how the samples are going to be used. You can have two types of samples. One is the saliva samples, mostly for the isolation of DNA, or you can also donate a blood samples that can use for do both isolation of DNA as well as the identification of circulating biomarkers. So that can be RNA proteins that can then be used to monitor progression of the disease, to identify therapeutic responses after a treatment, as well as disease progression. Those are all the people that we are funding. There are quite a lot, and if you have heard my presentation, that many of those investigators often wear multiple hats. We have Dr. Monco and Angela that is doing 2 or 3 projects simultaneously. Same thing for Mariam, item for Dr. Stella, and many other of the people that we are funding. I want to thank you because you are our biggest advocate, and there is a lot that you can do to help NDF helping you. And the most important thing is get involved, raise awareness about the disease. People do listen. And I can tell you stories of government agency like NIH that funded investigators simply because they were tired of hearing the patients and responding to the patient. Why are you not funding this? So make your voice heard. There is a lot that you can do. And reach out to the community and reach out to NDF. Make people know that you count, because you do. And if you want to help, please register. If you haven't done so yet, please register, because one of the most important things is we need to know how many patients. We need to be able to identify as many patients as possible. We know that the majority of the GNE myopathy patients are still undiagnosed, and we need to know, and we need to be able to know those patients so that we can eventually deliver a therapy to the entire community. I want to thank the members of the Scientific Advisory Committee. Here it's you can see who they are. I want to also thank the board of directors, because those are really doing most of the work behind the scene that we don't often see. And a special thank goes to Lale, who has been incredible at NDF. I've been in the subcommittee for the past 12 years, and I've seen the changes that Lale has been able to make at NDF, at the contribution, and how much more closer we are now to a cure because of the tirelessly work that Lale does every single day. And a big thanks also to Nancy and Rachel. And finally, thank you guys. You push us every single day because we know you. We're working harder every single day. So thank you all.
Thank you so much, Carmen, for that elegant presentation and the embarrassing show of confidence, but it's nice to hear. Thank you so much that you guys are pleased. Certainly scientists have definitely rallied around what we're doing over in the last few years. We've grown from a small group of scientists to a much larger, newer group of scientists, which is always good news for any rare disease to have people interested in working on it. Thank you so much for really summarizing for us all of the work that we are doing and filling in the gaps in our journey towards an IND in clinical trials. You know, a lot of people don't really understand the process of all of the work that has to happen before you are approved to experiment on human beings. And when we get closer to that valley of death, is it that you call it, Carmen, which is what a lot of people call the end of phase 3 before it goes commercial, that is where the real trouble begins. So, you know, getting scientists to dose it, but getting a biotech to agree on the vectors that are used because they're in it. Let's be honest, yes, they have a mission of coming up with drugs, but they also need to make money because they're for profit. So in other words, the selection of the vector becomes an incredibly difficult piece of that puzzle. And that is why at this point, where if you go back to that slide that you were talking about, the various tiers, which is also reflected in a small cartoon we made for, so between tier 0 and tier 1, which is where it's, you know, you begin that, and the drug approval phase, there's a lot of things that have to happen. And the more you understand about the process, the more you realize how hard it really is. And we are, I believe, doing all of the right things based on a consensus of scientists that maximize our opportunity not to fall through the gaps between T3 there and T4. Speaking of biopharmacy, Carmen, I just wanted to make one comment on your presentation about Ultragenyx. They, in fact, haven't completely abandoned our disease, GNE myopathy. They still have a prodrug, and every time I talk to Dr. Emil Kakis in passing, or his staff, or the chief medical officer there, they assure us that they are very interested in continuing work with that prodrug on sialic acid. In the comments section, I know that Tara has made a comment about how it was helpful to her, in fact. And we hear this from a lot of different patients. So there is going to be some discussion about why some of the sialic acid was effective for some patients and perhaps not as effective for others. And also, I just wanted to correct one other thing, that with regards to valid outcome measures, which we tend not to think we have, I just want to say that the NIH does in fact claim to have some valid outcome measures, and hopefully with the inclusion of Dr. Nuria Carrillo on our team, we'll be able to have either access to more information on that and maybe learn a little bit more. Do you have any comments on that, Carmen?
I agree with certainly the outcome measure that we have right now, they're not optimal. So we need to work on the outcome measure. We heard from the presentation that Marian gave that, yes, we do have some outcome measure, and probably the cialliation is the most effective one, but the more we can find, the better it is. And I know that NIH is working very hard at identifying those. Nuria, do you want to add anything to that?
Yeah, I'll be delighted because this is not yet published, but it will be soon. And well, some of it has already been published, but it was a mathematical paper, which was complex to understand. But the fact is that we actually do understand very well disease progression, which is one less thing that we need to worry about, and that the outcome measures for determining clinical efficacy are very well understood and actually good outcome, valid outcome measures. So again, one less problem we need to figure out. So I just wanted to mention that.
Yeah, thank you. And I really like particularly the model that NIH has of disease age rather than stage of the patient, of age of the patient, is the disease age. Correct. Which is because going to be critically important.
For in lay terms, the way I understand it is, so there's a difference between how long ago a patient was diagnosed versus how quickly that disease has progressed. Because one of the things that I know is a challenge for us in GNE myopathy is that for some patients, they can be walking with a cane for 25, 30 years after diagnosis, and yet others can be in a wheelchair within 5 years. So is that what you're talking about when you talk about disease age? Could you please clarify that for some people that may be listening who don't completely understand the subtle difference?
Sure, I will. So I don't want to go into many details, but when we refer to disease age is how much, regardless of the age of a patient, how much has the disease affected that particular patient? And so it's a measure of overall disease progression. That's all it means. Disease age equals the amount of disease progression a patient has had, regardless of their age. So it may have been 20 years since they had the onset of disease, but their disease age may not be high because they're progressing slowly. Or people that started with the disease very recently could have an advanced disease age despite the fact that only a small amount of time had happened. So it takes into account the individual rate of progression of each patient. So that's what it means.
Thank you so much for that explanation. Going to some of the comments and questions, and there's quite a few. Tara Google wants to say that she endorses and likes the designation of our various stages in disease age. She thanks you, Dr. Bertoni, for a wonderful presentation, and everyone is congratulating us and Nuria on this new role. There is a couple of specific questions. Patient asks, I may have asked this question before, but still don't understand. Do we know to what extent other genes are interacting with the GNE gene? If other genes are interacting with the GNE gene, do they play a role in the onset, symptoms, and potential recovery of GNE myopathy? And quite frankly, this is a question that we get often from other scientists to whom we try to present our disease.
Okay, then again, maybe Nuria can comment on this, but I can comment on what I know from other diseases like Duchenne muscular dystrophy, which has been one of the first to be identified. We do know that there are other genes that can influence the progression of the disease, and if this is true for diseases like Duchenne that affects very young children, it's very likely to be also true for other diseases, including GNE myopathy. I'm not aware of many people that have described new genes that could be involved in GNE myopathy or could influence the progression of the disease. We clearly know that GNE myopathy is due to mutation in the GNE gene, and that's a fact. Now, whether there are other genes that could also play a role simply because any of us has genes that influence how we do and who we are, that I think is not completely clear, and I hope Nuria can comment on this based on what she knows on from the literature.
Nuria, could you come back on and address that question if you're still available? No, she's gone.
Sure. And
No,
I don't have more information, but I just want to kind of explain the concept. All genes are constantly interacting with the product, the product of a gene constantly interacts with the products of other genes. And so, Carmen is right. There is not much in the literature that talks about what are the main genes that interact with GNE other than within the pathway. I think that some of the research that she was showing is being done may give us more insight into what are those genes that are playing a role around GNE. And people look for GNE modifiers. I think there is work being done already too as well, that it's funded by by NDF, that once those mechanisms start to become more clear to the scientists, then we'll know which genes are interacting with GNE. But as in other genetic diseases, of course, the function of GNE that's missing is dominating the lack of function in the system due to the disease. But
so thank you for that clarification. And so that our whole genomic sequencing study, or the study that, for example, Dr. Lex are doing at Yale, those are things that can show us perhaps the function of other modifiers and the role of different genes. Am I correct? Is there anything else that we're doing to try and answer that question? And what questions should we be probing?
Well, I think what you discussed about the membrane integrity in muscle diseases,
Dr.
that also can understand, I think, there is the role of GNE has with antioxidants may give us more. The reality is we don't know that much about GNE other than it's part of this enzymatic pathway. And that is, one of the things that scientists work every day to discover. And I think most people scientists in the scientific community would like to know more about that. We do know it's a very important gene, but we just don't know how exactly it connects from not being able to work properly to only leading to muscle deterioration. I don't think anybody understands the full connection between one and the other yet.
I see. And Carmen, did you have any follow-up comment to that?
No, I agree. We really, right now, what we are exploring is the possibility of using, for instance, blood samples from patients could be a very good way to discover new biomarkers or metabolites that could influence the progression of the disease, and could also be used as outcome measure. And I think we are at the forefront of the technology, and we are sponsoring study that, like the studies with Perkinamab, that had the potential to truly identify the biomarkers if they are present.
Right. And I think that's a kind of recap of all the things that we've been trying to explain to our donors and patients and all stakeholders, in that we really are trying to fill in the gaps and understand as much as we can before we start to dose human beings. And this is critical. There have been a couple of diseases that have gone on ahead of us with regards to gene therapy, and they have been really experimenting with the dosage without knowing perhaps everything that they can know. And it's a fine line, I suppose, to know when you know enough in order to start dosing, because there is such a sense of urgency among a lot of patients for whom my favorite comment from one of our board members and patients, Michael Banayan, is, time is muscle. At the end of the day, though, we really do have to be very careful and conscious of the fact that the scientific community knows enough about it, not just one or two scientists who may be mistaken, but a consensus of scientists who know enough about it to understand the role of modifiers, other genes, and therefore the amount of dosage and things, because as some people who are following this, there have been some missteps from which we hope to learn by the mistakes of others. So we are blessed in that regard. Thank you for that feedback. I have one final question from Christy Bowling, who is texting us and saying that she wants to know the difference between GNE myopathy and learning about myositis, as her husband has it and is currently undiagnosed. Can you explain the difference? Because we do get these questions a lot. A lot of people do come to us with different kinds of muscle disorders. Could you just briefly give us
I'm going to ask Nuria to respond this question, because she is the most appropriate to discuss patient and diagnosis. So Nuria, if you can.
Great.
Yes, I'm happy to do that. So in myositis is a group of diseases that affect the muscle due to inflammation. So that's very different from what happens in GNE myopathy. In GNE myopathy, there is no muscle inflammation. Things that can cause someone to have myositis could be like an autoimmune disorder, so the own body attacking the muscle. There are some other forms of myositis that may be genetic. There are another forms of myositis that can be caused, for example, by medications and things like that. So typically, one of the clues that we have to make the diagnosis for GNE myopathy is when we're looking at the muscle biopsy, there is no inflammation. So that is telling us already that this disease is not a myositis. So again, in terms of myositis, there is a very long differential diagnosis. And so I would recommend Christy to make sure that her husband is evaluated by a neurologist and an academic institution that will be able to figure out and do the necessary tests to rule out the different diagnoses.
Thank you for that clarification. And Nuria, I know that you're joining us officially on Tuesday to wrap this up. I just wanted to ask if you, Carmen, perhaps Dr. Tassine Mozafar, who's on our board of directors, Ralph Lauren, and some of our other scientists would be willing to do a panel discussion this way, so that we can have an interactive session the way we normally end our symposia. This is just an idea, but I'm wondering if you would be willing to participate in a discussion where everybody kind of hears the conversations that scientists have among themselves and can directly and perhaps interactively ask questions. Would that be something you both are willing to do?
Of course.
Thank you. Wonderful.
I need to make a correction to one of the things that I said. I just received a text. I mentioned that the gene was identified in 2001 by Dr. Argov. My apologies, is actually Dr. Stella Rosenbaum. And power to the women. So sorry about that. Please don't take it personally, as it was a lapsus. So thank you, Stella, for the hard work.
Thank you. And we have, for the record, we have multiple times promoted Stella Rosenbaum, who is a wonderful, true pioneer in this disease, has really been the woman who discovered this in the year 2000. And Dr. Argov was blessed by association, not to take anything away from Zohar there, but yes, it really was Stella, and we have multiple times said that, and Stella is a very, very big part of us. And actually, I think we should do a series on the women of NDF, including Stella. And half of us have accents and from different places, so I think it'll be a very interesting discussion, especially with the climate right now in this country, certainly. So thank you again for your participation. You'll be hearing from the rest of us. We are going to take a break from this series, everyone. September 4th is a holiday here in the United States. So we will therefore resume some discussion and announce any further talks by either Dr. Martin, by Dr. Nuria Carrillo, and a panel discussion led by Carmen Bertoni further. So do follow us on Facebook, and in the meanwhile, you will see our announcement on LinkedIn. So please do go and see that and post it and let people know that for a tiny little nonprofit, we are making a lot of noise. Mona, did you have something you'd like to, for those of you who haven't met Mona Patel, take you're muted, so turn on your mic. I wanted to introduce Mona Patel. She is our certified patient advocate out of the United Kingdom and works so hard to get our messaging out to the people in that part of the world. Mona, I'm assuming you have something you'd like to say.
I just thought we've got an opportunity to see each other, and I've been missing the social interaction during the pandemic. So I thought before we leave, could we all just see each other and say bye and just share and celebrate this wonderful news today?
I would love that. Go ahead and turn on your cameras, everyone who wants to be on. We would love to see you. And as a matter of fact, I was going to suggest, while you're all turning on your cameras, if you'd like to, I wanted to suggest that when we do, in fact, have a panel discussion, we can make it, we can go up to 250 faces. So please, anybody else? Nancy, do you want to turn on your camera? She's going to say no, I know it.
Not to put anyone on the spot, but anyway, I'm going to say bye. I'm going to get going. But I wanted to
say no. So here I am.
Anybody else? Amy, Tara, Rose, Michael.
Hello, everyone.
Valeria,
I would not turn on my camera because I'm just having coffee here.
Okay, we're all together.
My hair is not done. I'm in my PJs, so.
Well, it's really important, I think, for all of us to see. We have participation from, let's see, Rose Okama out there in Africa, Mona Patel from the United Kingdom, Michael Banayan from the United States, Valeria Pace from Italy, Falguni Mehta, I think from India, if I'm not mistaken.
East Coast.
I'm sorry, from where?
She's from the East Coast.
Oh, she's from the East Coast. Okay, but we have Daipan man right there. You're sideways. You are, where are you calling in from? I think India. I saw that. Anisa Osamu Sasaki from Japan. So you can see that we have what once started out as we believed that when this organization was founded in 2006, that this was a Jewish Persian mutation and a disease that was limited to that community. The good news is that we have discovered identified patients in 35 countries and counting, and that is good news. Why is that good news? Because, to be honest with you, if we want to find a biotech and a biopharma partner, they're going to want to know that there is in fact a head count for the drug development. They're not going to spend $50 million plus on something if they don't have customers across the globe. So this is all very, very good news. Thank you to everyone for participating, showing us your lovely faces. And next time what we'll do when we invite you on, we'll give you a heads up so you can do whatever you want to, take your hair out of the ponytail, add makeup, whatever you want to do. Thank you again. Welcome Nuria to our team and to the Inner Sanctum. Carmen, thank you so much for this lovely presentation. Nancy, Lurie, thank you for all you do every single day. And of course, Rachel is on vacation this week, but thank you to everyone. And we look forward to seeing you on the internet. As always, you'll see a recording of this on curehivm.org.